Hello to everyone. It's nearly 11. We're about to restart our conference after this break. Just waiting for a few seconds. Okay, it's 11. I can see that you've all connected. Nearly all of you have connected again. So as planned in our schedule, we're now going to have a pitch session about very interesting projects in connection with our session 3 about tools development. And to start, I can introduce Mr. Tom Sharrock. You are Artificial Intelligence Engagement Manager at Lifebit, and you're going to talk about real-time disease surveillance using artificial intelligence and Lifebit REAL. It's up to you now.
Thank you very much, and good to be here. Thank you to the organisers. So thank you for the kind introduction. My name is Dr. Tom Sharrock, and yes, I'm the Artificial Intelligence Engagement and Product Manager at Lifebit. And today I will be talking to you about how we're undertaking real-time disease surveillance using artificial intelligence and our software product called Lifebit REAL. So just to give a brief background on Lifebit for those that have not heard of us before. So we're a leading population genomics and precision medicine company that is powered by our federated platform called CloudOS. And on top of our federated platform, our technology platform called CloudOS, we have a variety of different applications which are powered by both federation, so the ability to look at data without it moving, and artificial intelligence. And we have 5 different applications, but in particular, today I'm going to be talking about our product, Lifebit REAL, which enables you to undertake disease tracking and discovery in the drug discovery and epidemiological space, and particularly for disease surveillance. So Lifebit REAL provides an up-to-date view of the current spread of infectious diseases and also provides information on disease prevalence for syndromes and non-communicable diseases. Lifebit REAL enables early detection and reporting of emerging agents, so novel viruses which have never been seen before, in addition to the transboundary spread of diseases across geographic regions or zoonosis. The key thing about Lifebit REAL is that it alerts users to outbreak events that we have detected earlier, so that you can basically get a preemptive warning and for early disease surveillance. The way we do this is by ingesting internet data and combining data which is found on the internet with artificial intelligence. And I'll speak a bit more about that in a moment. But first, I'll just highlight one of our partnerships is with Boehringer Ingelheim Animal Health, where we have combined with them to basically undertake global disease surveillance for the animal health sector. So Lifebit REAL works in a 3-step process, that is ingest, extract, and report. So firstly, for ingestion, we basically harvest articles from the World Wide Web, scientific sources such as journals, social media, and news outlets, and we do this in a real-time basis so that we get the most recent articles that are up to date. The second step is an extract, and here we use our proprietary machine learning and natural language processing algorithms to extract data points from these articles, and we basically look at disease and outbreak mentions in an automated way. And finally, we then report this. So disease and outbreak information is compiled to reveal insights into disease prevalence and infectious disease spread. And we provide custom email alert notifications for when diseases and novel agents are identified, which are of interest. So to dive into the ingestion step in a little bit more, we look at data sources such as scientific journals, abstracts for conferences. We look at public health and government organizations as well. That's alongside universities. But one of our key differentials is that we look at news outlets. So these are both global media organizations, but in addition, regional news outlets, which are often actually a great source of information for disease outbreaks very early on during an outbreak. And in addition to that, we also look at social media and blog posts. And these are very valuable, especially for companion animals, because a variety of different pet owners will talk about their animals online and mention if a dog has a particular disease or if a feline has a problem. The second step is an extract. And what we do here is use natural language processing to turn unstructured data, which you can see here, a mention of avian influenza in Russia, and we turn that into a structured data point by using natural language processing, And we build up these data points within our system so that we have a global view of disease spread and disease prevalence. And finally, we have the report module. So here we synthesize all of the data that we have gathered together, and we can then report when emerging viral agents are mentioned within the text and when diseases which are known to be infectious spread around the world. In addition to that, we can provide data points on market growth and disease trends, and also help organizations identify the best place to have a clinical trial where endpoints are likely to be reached due to a high prevalence of a disease. Now, to give a bit of a use case, we can actually track the UK norovirus spread this year in the UK. This is obviously a little less mentioned in the news and media compared to COVID-19, but here's a great example of how you can look at data which has been gathered from government agencies, from news outlets, and also social media to basically start building a picture of the spread of a disease within a particular geographical location. And what we're able to do is actually map the prevalence of these diseases for both human infectious diseases, animal diseases, and also non-communicable diseases. And here you can see some good examples of different Reddit posts, for example, also government organizations, and also Twitter posts for norovirus mentions within the United Kingdom. And this can basically have the same use case on a variety of different other diseases. So that's all I wanted to talk about on disease surveillance, but another part within our Lifebit REAL application is our drug identification module. And here what we were aiming to do, and this is more in an R&D phase, is actually identifying the drugs and vaccines which are in development for a particular disease which we have tracked. And by doing this, we can start to actually build a knowledge graph, which is a variety of different data points connected using artificial intelligence, so that we can start ingesting data from that, be it scientific research articles, scientific review articles, or even tweets by an academic scientist. By looking at this data, we can start building a picture of disease prevalence and disease surveillance for these diseases. And in addition to that, we can also then look at the drugs, such as drugs mentioned here on for these particular diseases, and the genes and proteins which are important, and start building a knowledge graph of both disease, drugs, and genes. And we do this in real time and enables companies to basically understand which drugs can be used and are in development for which diseases. It provides an understanding of mechanisms of action for these diseases as well, and also enables in-licensing opportunities and novel uses for drugs that have previously not been thought of before. So thank you very much. I appreciate this is a very short pitch, but if there are any questions and anybody would like to connect, my email is tom@lifebit.ai, and it's been great to present to you today, and thank you for listening.
Thank you very much, Tom Sharrock, for this presentation, which was brief and very clear. So now let's switch with Cyril Guyard. Cyril Guyard, you are Deputy Chief Scientific Officer and Director of Technologies at Bioaster, and you're going to talk about One Health tool development within an integrated microbiology technology institute. It's up to you. We can't hear you. I think you should connect your mic. Turn on your mic. It's a green mic on your computer. No, we can't hear you. Okay.
We got it, yeah? Yeah. Good morning, everyone. Just first of all, I'd like to thank the organizer for inviting Bioaster to make this presentation. That's a great honor for us. And as you just mentioned, my name is Cyril Guyard, and I'm Deputy CSO at Bioaster and Director of Technologies. So let's switch to my presentations. Okay, I have a control now, eh? No, I don't. Can you give me the control?
Yes, you do. Simply click.
Okay, got it. Yeah, thank you.
Thanks.
So just like I will just start quickly with a short introductions of what is Bioaster, and then later on I will just give you some overview of what we do, the field we're covering in terms of One Health approach and technology that comes with it. So Bioaster is a not-for-profit institute of technologies. We're located in France, in Paris, and in Lyon, and in Paris on the Pasteur Institute campus. We're established as a foundation for scientific cooperations, and we are tackling a novel approach for R&D based on integrations of major scientific and technological pipelines. We're focused on the microbiology and infectious disease. Our team, we're like about 100 people, more or less, and most of our staff are scientists that are coming from all over the world, with a major idea to integrate multiple skills at the same place. And we have a chance to have the support of high performance computing and data storage facility, as it was mentioned before during the roundtable. I mean, tackling all the questions we're trying to answer, and requires to have access to high power computing. And for this, we have the support and help collaborations of CNRS, CC, IN2P3 that is listed here. So we're addressing the four field of applied microbiology. I won't go into details of those the four field that are described here in terms of where we applied our energy, but basically we're providing answers for antimicrobial development, vaccines, diagnostics, and microbiota. So since our creations, and we were created in 2012, we managed to obtain and agree on 250 research contracts. Most of a large part of our activities in terms of volumes is dedicated to our joint research project with the industry, with a total number, and academics, with a total number of 62. We're basically turned toward collaborations, as you can see from those numbers. We ended up working up to now with 93 different industry partners and 54 academic partners. And we have our own technologies that we brought to maturity so they could be used in the industry field. So just another view of all the technologies and teams we have gathered at Bioaster. We're basically pushing mostly on the integrations of those technologies and teams, which are basically in the order from left to right: clinical microbiology and molecular engineering, development of preclinical models, and imaging strategy for those models. I will give you an examples later on in terms of technologies and tool development. Bioassay and microsystems and optical engineering. Here we're basically talking about optics systems, about also microfluidic systems, also spectroscopy analysis, and computational microscopy. A large part of our activities is dedicated to omics, and we're basically covering most omics here in our field of interest. That comes also with a lot of work, obviously, with bioinformatic pipelines and biostatistics. A large part of our attention is also turned toward data management and data science, because it's obviously a big challenge right now to gather, integrate all these informations when you're talking about all those diverse omics data. Here, this is just the first examples of a project that is ongoing at Bioaster, where we're looking at cross-species omics integrations. This is here developed in the spirit of a therapeutic product development, but also prophylactic product development. Obviously, you could basically imagine the use of such strategies in a One Health approach. Here, we basically are looking at developing therapeutic prophylactic solutions, where it is important to have access to cross-species response to therapeutic solutions. It comes with obviously strong and difficult ability to compare the reactions to a molecule or a syrup or prophylactic solutions across species. So we are performing a large amount of work on the autolog comparisons. So once those autolog comparisons is performed, you can then integrate clinical, preclinical data with omics and metadata. This comes with challenges for aggregation of those data, multi-omic integrations, and after that, biologic interpretations across those different species. So you can basically correlate those events in those different animal species. Here we're talking in terms of animal species of rabbit, NHP, non-human primate, and mice. The second part of my presentations will be on, obviously, this is needless for me to say that the One Health approach can contribute to the AMR threat we're all facing. Part of the answers can come from microbiome particularly, and this will be the focus on the last part. And those requires tools for the analysis of microbiome resistance, but also alternative solutions to identify the antimicrobial of tomorrow rather than the classical antimicrobial. In this case, we're employing two strategies. One that is on the metagenomic side that we all know from the analysis of environmental microbiome, but also culturomics. Culturomics, in this case, we apply it to new antimicrobial, but also on the discovery of new live biotherapeutic product. For the metagenomics, we are basically focused on the microbiomes, certifications, and resistance analysis. Oops. Here, first example of a technology we've developed. We all know the bias when it's about analyzing the microbiome. We all know the bias of current solutions, which are focused on 16S RNA sequencing, which are mostly with a mostly used one, the V3, V4 areas for sequencing. Here we've developed solutions to sequence from V1, V9, basically V3, V4 sequencing, 16S RNA sequencing, basically has the limitations of not being able to go down to species in terms of analysis of a microbiome. Here we've developed a tool that provides you a full picture of V1, V9 with reduced error rates in our case for the technology we've developed, and with specific methods, we are able to go up to 100% of sequencing with differentiation of species, where the classical methods go with 73.5% of identification of species with V3, V4. I invite you to go to look at to take a look at the. Oh, I get only 1 minute left. Okay, thank you, Fabrice. So take a look on the YouTube. We got information about this. Okay. Another solutions is integrated pipeline for generic analysis of shotgun. Here we're more talking about the software development. Please take a look at that. That's a really interesting solutions. We are tackling also the solutions for therapeutic solutions. The second part on the right side of my initial graph. Here we've been looking at in collaboration with the Ferricides as biodistribution solutions for phage therapy analysis, and now we're looking at efficacy. Not talking about topical treatment, but about systemic treatment here. Last solutions, the culturomics, classical one. We work with Boehringer Ingelheim in this case, and we could isolate with classical culturomics anti-campylobacter activity for in vivo and in vivo administrations. Next generations is this ones. We now have a pipeline to do next generation culturomics, automated anaerobic conditions. And with this case, we can really speed up our discovery of new probiotics. And I'm going to stop here. And please feel free to take a look at all our collaborations and links, so you could get more information of our technologies.
Thank you very much, Cyril.
It was super fast. I'm sorry if I was.
Yeah, no problem, no problem. I'm sure that people will contact you to know more or to talk with you about your wonderful project. Thank you very much for participating. Thanks. Now it's time to talk with Julien Flaig. Julien, you are founder and the managing member of Epimod, and you're going to present solution Epimod.
Hi, thank you, Fabrice, and good morning, everyone. As you mentioned, I'm working at Epimod. I'm doing mathematical epidemiology in the company. Epimod is a small company providing applied research services in the field of epidemiology of infectious diseases and public health. We have 5 staff members with skills in statistical and mathematical modeling, public health, field epidemiology, but also human resources and healthcare systems. We have been working on various infectious diseases like bronchiolitis, also some STIs like chlamydia, and also malaria, which will be the focus of my talk today. Malaria is an infectious disease caused by Plasmodium parasites that are transmitted to humans by infected Anopheles female mosquitoes. So the transmission route goes like this. Mosquitoes bite infected individuals, they get infected, and they can transmit the parasite to healthy individuals. Malaria is still today a major cause of mortality in some low-income and tropical countries, especially among young children under the age of 5 years. But malaria is also a very good example of a One Health issue. And this for 2 reasons, 2 main reasons. The first one is that not only humans, but also livestock can get infected. And the second reason is that mosquitoes need water to breathe, which means that the environment plays a major role in transmission. So some interventions will try to protect humans directly from getting bitten by mosquitoes or getting infected by the parasite. This is the case of the vaccine, for instance, or repellents or bed nets. Other interventions will try to destroy mosquitoes' breeding sites, that is, change the environment. And this is interesting because changes in land use can have very complex implications and sometimes even adverse effects. There are also interventions that try to control mosquitoes more directly by simply killing them or introducing sterile male mosquitoes. And finally, some livestock-based interventions have also been proposed. For instance, using livestock to distract mosquitoes from biting humans. And this is called zooprophylaxis. And this strategy is a bit controversial because the exact opposite effect has also been reported, namely that the incidence of malaria may in fact increase in the presence of livestock. And one way to cut the knot is to use insecticide-treated livestock to attract and then kill mosquitoes. Importantly, individual behavior are very important for all these interventions to succeed, which makes malaria control also a very complex social and political issue. You see also that there are many possible malaria control interventions, and these interventions can be combined together, which means that the number of possible policies that need to be assessed in decision-making is very large. In addition, control policies can be defined dynamically. So they can change over time, you can change interventions month by month or week by week. And interventions can also be defined spatially. For instance, implementing one intervention in a given region and no intervention in a neighboring region or a different intervention. So this makes the number of possible policies very huge. And in addition, malaria transmission is a very complex and non-linear phenomenon with many uncertainties. All these factors taken together make the use of mathematical modeling and numerical simulation unavoidable in decision-making. Mathematical modeling can be used, of course, to anticipate or try to anticipate the impact of one single intervention or combination of intervention. It can be used to compare a predefined set of interventions, so typically a small number of interventions or combinations of interventions that are deemed relevant. It can also be used to do optimization. Optimization means trying to find the best policy among many policies without assessing them one by one, because this would be impossible. And finally, numerical simulations can help to deal with uncertainties by running many simulations of uncertain scenarios. Of course, mathematical modeling is impossible without quality data. And here I would like to give more details about the example of data collection in Togo. So you see Togo here in yellow on the map on the west coast of Africa. Epimod has been present in Togo since 2016, and a local subsidiary was founded in 2019. The Togolese health system is relatively well equipped, at least when it comes to data collection, which makes it easier to collect various data. Of course, surveillance data, that is cases, malaria cases, also entomological data regarding mosquitoes, parasite data regarding Plasmodium parasite, and also weather data, because the rains have a role in malaria transmission. The cornerstone of the system are community health workers. They are volunteers whose job is to report malaria cases on a monthly basis. The surveillance data was used in 2 PhD projects supported by Epimod so far. The first one is a project by Francis Bakai. It was supported in September last year. And the second PhD project by Anne Thomas will be defended in 2022, this year. So there is data available in Togo. However, the country still has one small weak spot, which is the lack of locally trained scientists at master's and PhD level. In the next few years, our objective will be to try to contribute to this virtuous cycle by supporting the training of scientists. And we hope that they will produce quality scientific results, good scientific results based on quality data, and that this will attract in turn new partners and new funders, for instance, pharmaceutical companies who might be interested in carrying out clinical trials in Togo. We have already started this work with the 2 PhDs I mentioned. Our current projects are mostly data analysis projects. We also have one project aiming at improving the surveillance system in the country. And in the future, we want to carry on in this direction with more and more qualitative analysis of malaria in Togo, modeling and simulation projects, working on improving the healthcare delivery system, so intervention delivery, and finally, carrying out clinical trials of interventions. This concludes my talk. if you want to reach out, you can find us very easily on LinkedIn, or you can write me an email directly at the following address, jflaig@epimod.fr. Thank you for listening.
Thank you very much, Julien. And I think, noted your mails and contacts for us to continue and maybe talk with you about your project. Thanks a lot for participating.